If you find yourself looking at a diagram like this one of an animal cell and thinking to yourself, how am I going to master all of this material? If you're feeling overwhelmed and nervous, then this video is for you. Here's what we're going to cover. We're going to start with an introduction to cells, then we'll look at why cells are small. We'll follow that by looking at how cells are internally organized. That's about cell compartmentalization and a system of organelles that are organized into an endomembrane system. After we do that, we'll be able to walk through cell parts and functions. Then we'll look at membranes, how membranes are made up, how membranes transport things from one side to the other, and finally, we'll look at osmosis. My name is Glenn Wokenfeld, also known as Mr. W. I'm a retired AP Biology teacher. I love biology. To help you study, I've put together a checklist that you can download at apbiosuccess.com/checklist. Cells and introduction. Describe the basic structure and fundamental parts of cells. Cells are the basic unit of life. They're the basic unit of structure and function in every organism. In terms of their own structure, they have a membrane which separates the cytoplasm, all the living material inside the cell, from the cell exterior. They always have genetic information in the form of DNA, the double helix, and there are systems that maintain and replicate the cell as a whole and the cell's genetic information, its DNA. One of the most important systems is a system by which the genes in DNA get transcribed into messenger RNA, another nucleic acid, and that messenger RNA is read by a particle called a ribosome and translated into proteins. Proteins are all important. They're the workhorse of the cell, and one of the most important proteins are enzymes. Enzymes control the cell's metabolism. Other proteins might be embedded in the cell's membrane, and others might even be exported from the cell. Compare and contrast prokaryotic and eukaryotic cells. Among living organisms, there's a great divide between the prokaryotes and the eukaryotes. Here's what it's all about. Prokaryotic cells are small and relatively simple. They have no nucleus. Their chromosome is circularized. And what that means is that there's a loop. The beginning and the end meet up. They contain extra chromosomal pieces of DNA that are called plasmids, and they're found in 2 of life's 3 domains, the archaea and the bacteria. Eukaryotic cells are larger and more complex. They're found in the domain that has their name, the eukarya. They have a nucleus. They have multiple linear chromosomes. So here they're shown over here, and you can see the chromosomes here. They have a beginning and an end. And those chromosomes have DNA that's associated with proteins, which is different from the simpler DNA packaging that is found in the prokaryotes. Eukaryotes have mitochondria, and the presence of that organelle is the defining feature of the eukaryotes. And they have many membrane-bound organelles. Membrane-bound means surrounded by a membrane. Cell size, an essential topic. Use the relationship between surface area and volume to explain why cells are small. Cells need to have enough membrane surface area to allow for diffusion of substances and molecules in and out. Everything that gets into a cell gets there through the membrane. And it might be counterintuitive, but a small cell has much more surface area relative to its volume than a large cell does. Let's look at the math. If you have a cell that's 1 micrometer, 1 micron on the side, that's its length, then its surface area, if it's cubic, would be 1 times 1 times 6. Think of the formula for surface area for a cube. Its volume is 1 times 1 times 1, and therefore its surface area to volume ratio is 6 to 1. 6 units of surface area to every 1 unit of volume. Now, if you have a larger cube, one that's 10 micrometers, then its surface area is 600. Wow, that seems like a lot. But its volume is 1,000 because volume's a cubic function and it's going up much faster. So if you look at the surface area to volume ratio, it's 0.6 to 1. As an object gets larger, its amount of surface area relative to its volume decreases. And in this example, the larger cell's surface area to volume ratio is 1/10 that of the smaller cell. And a large cell can't efficiently use diffusion to get the nutrients it needs in and to get waste, carbon dioxide and metabolic wastes out. Why do organisms need to increase surface area in certain tissues? How do they do that? The why is in order to increase the surface for diffusion of molecules or heat. How? You can have thin sheets of tissue, like these thin sheets that make up the gills of a fish that have been dissected out. That's for increasing the intake of oxygen and the diffusion out of carbon dioxide. Or if you think about the ears of elephants, those are big, flat sheets of tissue that enable the elephant, which has very little surface area relative to its volume and lives in a hot place, to get heat out of its body. You can also have highly folded surfaces, like the internal membrane of the mitochondria or the structures that are called villi that make up your intestine. And there's actually folds within folds. Explain in terms of surface area why there are no small mouse-sized marine mammals, and why some marine mammals, like the whales, the largest organisms to have ever evolved, are really huge. Mammals are warm-blooded. Ocean water is cold. Increased size decreases the surface area to volume ratio, so there's less heat lost to the environment. Think about a mouse. It's small. It has a lot of surface area relative to its volume. It would be over here on the graph. It couldn't survive because it would experience so much heat stress in the water. The smallest marine mammals are the otters, and they have a thick coat of fur that helps them maintain their body temperature. Whales have evolved to be larger and larger for a variety of reasons, but one of which is that by being big, they lose relatively less heat to the environment than a smaller animal would. It's all about the surface area to volume ratio. Cellular compartmentalization and the endomembrane system. How eukaryotic cells are organized. What is cell compartmentalization? What are its advantages? Compartmentalization means internal division of a space into sections. Think about the overhead luggage compartment in an airplane. What are the advantages? The first is that it allows a cell to have regions with an internal chemistry that are distinct from the cytoplasm. A great example of that is the lysosome, shown at G over here and number 7 over here. Lysosomes contain hydrolytic enzymes. Those are enzymes that break things down. Those can't be released into the cytoplasm, or they would break down the cytoplasm. So they are compartmentalized. They're kept in a membrane-bound space. In addition, compartmentalization provides a lot of internal surface area, and that's important for membrane-bound enzymes that are found in the smoothie R or the Golgi that can only do their job when they're embedded in a membrane. Compartmentalization makes that possible. And that's also true of the ribosomes that are found in the rough ER. Compare compartmentalization in prokaryotic and eukaryotic cells. Prokaryotic cells have few compartments, though there are internal regions with specialized structures and functions. Here's a prokaryotic cell. You can see that its internal structure is much simpler than that of a eukaryotic cell. But one notable compartment is called a thylakoid, and it's this over here in a cyanobacterium. Cyanobacterium evolved into chloroplasts. We'll talk about that in a moment. And they have these internal compartments called thylakoids that are essential for the function of photosynthesis. Eukaryotic cells are highly compartmentalized. There are many internal membranes that divide the cell into regions with distinct structures, distinct chemistry, and functions. And examples include lysosomes, the endoplasmic reticulum, the Golgi complex, and vacuoles like this large vacuole in a plant cell. What is the endomembrane system? It's a dynamic connected system of internal membranes and compartments. It includes the nuclear membrane, the rough ER, the smooth ER, the Golgi, lysosomes, and all the little vesicles that interconnect them. Membrane and material flow from one compartment to the next. So the membrane that's part of the rough ER might become the same membrane, the same phospholipids that are part of the smooth ER, and those might become parts of vesicles which flow into the Golgi. So those phospholipid molecules are the same. It's dynamic, there's constant exchange. Explain the origin of mitochondria, cellular compartmentalization, and chloroplasts. What we're explaining is the origin of the eukaryotes. When did this happen? About 1.8 billion years ago. Before that, there was nothing on Earth but prokaryotes. How did this happen? It arose as a mutualistic endosymbiosis. Symbiosis means living together, two species living closely associated with one another. Endosymbiosis is when one of those species lives inside the other. We normally think of that as parasitism. That's a negative kind of endosymbiosis, but there's also positive endosymbiosis. That's a mutualistic relationship, and that is what gave rise to the eukaryotes. So what happened is that here we have representatives of the two prokaryotic domains, and an archaeal cell took up a bacterial cell. Now I want to emphasize that there are many scenarios and many diagrams that represent how this happens, and this one is the one that makes most sense to me. So the bacterial cell entered inside the archaeal cell and it evolved into a mitochondrion. That mitochondrion, still kind of a bacteria, secreted vesicles, and those vesicles became the nuclear membrane, shown over here, and it started to develop structures that are part of the endomembrane system, like the endoplasmic reticulum, which is shown over there. So that's how we have eukaryotes. Then later, a second endosymbiotic event led to chloroplasts, which were free-living cyanobacteria, entering into a eukaryotic cell, and that set the stage for algae and plants. What's the evidence that chloroplasts and mitochondria were once free-living bacterial cells that arose through endosymbiosis? What's the evidence for this idea? Like bacteria, both of these organelles, mitochondria and chloroplasts, have their own circular DNA. They replicate themselves through binary fission. That's how prokaryotes replicate themselves. They use their own ribosomes to produce some of their own proteins. These ribosomes resemble bacterial ribosomes in terms of their rRNA sequence and structure, and they have two membranes. The outer one, so the outer bacterial membrane, let's consider it over here, is a vestige of an endocytotic vesicle. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course. The material is complex, the vocabulary is ridiculous, and the pace is withering. It's natural to feel overwhelmed and inadequate. To get an A or a 4 or a 5, you need an easier way to study, and that's why we created learn-biology.com. It has quizzes, it has flashcards, it has interactive tutorials about every topic in the AP Bio curriculum. It has a comprehensive AP Bio exam review system. Use learn-biology.com and you'll gain the skills and confidence that you'll need to ace your biology course and to crush it on the AP Bio exam. So here's your plan. Go to learn-biology.com. We've got free trials from June through March for both teachers and students. You won't believe how much you'll learn. Eukaryotic cell parts and functions. Describe the structure and function of the nucleus. The nucleus's function is to store and protect genetic information in the form of DNA. That DNA is wrapped around proteins to form chromosomes. This representation here is only present during mitosis or meiosis when cells are dividing. Otherwise, the DNA is more diffuse and spread out. It's still in chromosomes, but we call the form chromatin. The nucleolus is this dark part in the center of the nucleus, and it assembles ribosomes. The entire nucleus is surrounded by a membrane that separates the chromosomes from the cytoplasm, and the nuclear membrane has pores that allow molecules to enter and leave the nucleus. Important among those are messenger RNA, which leaves the nucleus, goes into the cytoplasm, where it's transcribed into proteins, and various transcription factors that come into the nucleus to turn genes on or to turn them off. Describe the structure and function of ribosomes. Ribosomes are particles composed of ribosomal RNA and protein. They consist of a large subunit and a small subunit. Their function is to read a genetic message encoded in a sequence of messenger RNA and to translate that message into a sequence of amino acids that make up the primary structure of a protein. The details of translation, protein synthesis will occur in Unit 6. What are two locations within a eukaryotic cell where ribosomes can be found? Ribosomes can be free or bound. Free ribosomes, shown at 3, float freely within the cytoplasm, which is number 5. Bound ribosomes, shown at 4, are connected to the membrane of the rough ER. All ribosomes start out as free, though through a process called protein targeting, they migrate to the ER to become bound. They do that if the proteins that the ribosomes are creating are destined to be put inside a vesicle so they can go to the Golgi or the membrane or a lysosome. Describe the structure and function of mitochondria. Mitochondria are shown here in the context of an animal cell. Here's a larger image. The function is converting food energy into ATP. That's the molecule that cells use to get work done. The key structures, there's a chromosome that consists of DNA. There are also ribosomes, which aren't shown in this diagram. Evidence of endosymbiosis. There's an inner membrane. It's highly folded. That increases surface area. And that's because there are many membrane-embedded enzymes and proteins, one of which is shown at 5, that are involved in the process of ATP synthesis. The mitochondria, as a former independent cell, has its own cytoplasm. It's called the matrix. It has enzymes for the Krebs cycle and enzymes that perform other functions. There's an intermembrane space, which is an important cellular compartment that enables the mitochondria to produce ATP. We'll see how that works in Unit 3. And there's an outer membrane that is a vestige of endosymbiosis. Describe the endoplasmic reticulum, the ER, and list its two forms. The ER is an interconnected series of channels found between the nuclear membrane over here and the Golgi body in eukaryotic cells. So here's the ER, and it consists of two forms. There's the rough ER and the smooth ER. Describe the structure and function of the rough and smooth ER. Well, the rough ER is studded with ribosomes. That's what all these little dots are over here. And using those ribosomes, the rough ER synthesizes proteins for inclusion in lysosomes, in other organelles, in the membrane, or for export from the cell. The smooth ER is on the outer side of the ER network, always towards the Golgi, and it lacks ribosomes, but that doesn't mean that there's nothing in that membrane. It has many membrane-embedded enzymes, and the functions of the smooth ER vary by tissue. So it can include synthesis of lipids, converting toxins into soluble forms that can be excreted from the body, carbohydrate breakdown, and synthesis. Describe the structure and function of the Golgi complex. The Golgi is a series of membrane-bound flattened sacs, and you'll remember that flattening increases surface area. The Golgi receives vesicles from the rough and smooth ER and chemically modifies their contents. So here's the rough ER, here's the smooth ER, here's the Golgi, and here's a vesicle bringing something from the ER system to the Golgi. The Golgi packages those modified proteins into vesicles, shown at 6, that are sent to organelles like lysosomes over here at 7, to the cell membrane, or exported from the cell. Describe the structure and function of lysosomes. Here's a lysosome over here, here's a lysosome over here, and one thing that I rarely say, but I'll let you know that you will almost certainly never have to identify a lysosome on a diagram on the AP Bio exam. It's just a big vesicle. Lysosomes are membrane-bound organelles that contain hydrolytic enzymes. They're only found in animal cells. They carry out intracellular digestion. So like, for example, Here you see a particle. It actually looks like a cell that's being engulfed by the membrane. It'll be packaged into a vesicle, and that vesicle will be sent to a lysosome where hydrolytic enzymes will break this down. The lysosome also recycles worn out, damaged, or excess organelles and molecules, and it plays a key role in a process called apoptosis. The P is silent. That's programmed cell death, and we'll talk about that later in the course. The cytoskeleton is a dynamic network of protein fibers, and those fibers make up the bones, the skeleton of the cell. In the same way as our skeleton, our bones, working with muscles enables us to move, the cytoskeleton is a dynamic network that enables the cell to move both internally, so it enables cells to move materials and organelles within themselves, and it also enables the cell to move their membrane so that they can do things like endocytosis, where cells can engulf things by moving their membrane, or also moving their entire bodies. This is an organism called an amoeba, and it moves by extending parts of the membrane over the surface, enabling it to crawl. How does it do that? Through the cytoskeleton. What are centrosomes and centrioles? Centrosomes are the organelle. They contain 2 centrioles, and the function is creating spindle fibers, that's what these are over here, that separate chromosomes during mitosis and meiosis. The parts that we've talked about so far have all been represented within animal cells, but most of those parts are also found in plant cells. The one exception is the lysosome, unique to animal cells. Let's describe the function of the central vacuole, this large structure right here. It's only in plant cells. Its functions include water storage, storing and releasing macromolecules, sequestering waste products, and maintaining turgor pressure. We'll talk about this in the context of osmosis, the movement of water, but as water flows into a cell, it creates outward pressure that keeps plant cells full, firm, upright, they're not wilting. Describe the evolutionary origin and function of chloroplasts. Here are chloroplasts over here. Here's a single chloroplast. Chloroplasts are the endosymbiotic descendants of free-living photosynthetic bacteria, and that's why they have their own DNA, their own ribosome. That's why they have 2 membranes. Their function is creating carbohydrates through photosynthesis. We're going to leave their detailed structure and how they work for Unit 3 when we discuss photosynthesis. Describe the chemical composition and the function of the plant cell wall. The cell wall is composed primarily of cellulose, which is a polysaccharide. It's that polysaccharide that we can't digest, but which ruminants, cows, goats, can digest because they have these symbiotic relationships that enable them to break that down and release food energy. The major function of the cell wall is it acts as a pressure vessel that prevents overexpansion in response to inward flow of water, osmotic pressure. So here we see as water flows into a plant cell, the central vacuole will expand and it'll look like this. The cell will be full of water, and that water will push against the cell wall, keeping the plant cell full and firm, avoiding wilting. So that's a good thing for the plant. And the cell wall is the primary component of wood and the water conducting tubes in plant stems. Membrane structure and function. What is the function of the cell membrane? The cell membrane separates the highly organized contents of the cell from the cell's environment. Cells are open systems. They need to let things like food in. They need to let things like wastes out. The membrane is how that's controlled. That's because the membrane is a selectively permeable boundary. It allows the passage of some substances and particles through, but not others. That selectivity is really the key to life. Describe the structure of phospholipids and their role in cell membranes. Phospholipid structure, there's a hydrophobic nonpolar tail region. Here's the structural formula. Here's a representation. There's a hydrophilic polar head. Notice that the head has this phosphate group over here with its negative charge. The whole thing is held together by this 3-carbon molecule called glycerol. In solution, when phospholipids are mixed with water, the heads of the phospholipids bond with water molecules while the tails form a water-free zone. So interacting with water, water-free zone over here. That creates a bilayer. That's the basic framework of the membrane to which proteins and other molecules are added. We'll see that further on. And the phospholipid bilayer is stabilized by weak bonds, van der Waals bonds, that occur between the tails. Describe the fluid mosaic model of cell membranes. This model posits that the membrane is composed of phospholipids plus proteins plus cholesterol in motion. So why is it called fluid mosaic? Well, it's fluid because the components are moving around laterally within the plane of the phospholipid bilayer, which you can see here. It's a mosaic because it's composed of a variety of pieces in the same way as a mosaic tile is composed of different tiles with different colors that get put together to form the art. Within the phospholipid bilayer, you have phospholipids that are at 4 and 9. You have proteins that are at 1, 3, 5, and 6. And you have molecules like cholesterol, which are shown at 7. And then inside and outside the phospholipid bilayer, you might have additional molecules attached to proteins or phospholipids. And those might be glycoproteins, which are not shown, but at 8 you have a glycolipid. Glyco means it's a carbohydrate and it's attached to these fatty acids right over here. Describe how proteins fit into the cell membrane. There are 3 ways that proteins can fit in. And if you think about hydrophobic, hydrophilic, what binds with what, it makes a lot of sense. So there are transmembrane proteins. That's shown over here and over here. Here, notice that beautiful alpha helix that you should remember from our Unit 1 review. There's a hydrophobic core that fits into the nonpolar inner portion of the membrane. So these would be hydrophobic amino acids over here and over here. And there'd be hydrophilic regions that extend into the cytoplasm and the membrane exterior. There are integral proteins, such as the one at 5, and these proteins would have a nonpolar region that extends into the hydrophobic membrane middle and a single hydrophilic region that juts into the cytoplasm, as this one does, or into the cell exterior. And then finally, you have peripheral proteins, like this one that's shown at 3, and they attach to phospholipid heads that are either on the cytoplasmic side of the membrane or in the cell exterior. These terms, transmembrane, integral, peripheral, they're not important in and of themselves, but you should be able to understand the concept of how things fit into the membrane. Membrane transport, how things get across the cell membrane. Diffusion is the tendency of molecules to spread out from where they're more concentrated to where they're less concentrated. So here you have some substance more concentrated over here, less concentrated over here. Here's ink going into a beaker, more concentrated, and then less concentrated. This happens spontaneously. It's based on the kinetic energy of the molecules. And a good way to envision this is to think of molecules as flowing down a concentration gradient from higher concentration to lower concentration. Explain how cells control what diffuses across their membranes. Diffusion is called passive transport, and that's because it requires no cellular energy. There are 2 forms. The first is simple diffusion across the phospholipid bilayer. That's what's shown up here. There are 2 things that can do simple diffusion. The first are any small non-polar molecules such as oxygen, nitrogen, carbon dioxide. The second are non-polar substances such as steroid hormones or fats. Facilitated diffusion is for polar molecules and ions that can't diffuse across the phospholipid bilayer. So they require protein channels. And these are transmembrane proteins, they go all the way through, that only let specific molecules or ions pass. So over here you see one for glucose, over here you see one for amino acids, those are both polar molecules, and here you see one for an ion such as calcium. There'd be others for ions such as sodium or potassium. Compare and contrast active and passive transport. As you do, explain what powers each process. Passive transport is what we just discussed. It's when molecules or ions flow down a diffusion gradient. Here, more concentrated to less concentrated. And that's what it's all about. It's diffusion from high concentration to low concentration. It relies on the kinetic energy of the diffusing molecules or ions. It doesn't require cell energy. As you can see here, molecules are flowing down their concentration gradient. That's different from active transport, where we're pumping a molecular ion up its concentration gradient from lower concentration to higher concentration. Over here, lower concentration to higher concentration. That requires an energy expenditure on the part of the cell. Usually that's ATP that's being converted into ADP and phosphate to power the pumping process, but also the flow of electrons can power the pumping of protons, and that's important in the creation of ATP itself. Compare and contrast endocytosis and exocytosis. These are both forms of bulk transport. In endocytosis, the membrane pinches in to surround a particle or some extracellular fluid, creating a cavity that can become a vesicle. In exocytosis, a vesicle containing something fuses with the membrane, and then that contents is dumped outside the cell. Both of these processes require energy on the part of the cell, and they both require the involvement of the cytoskeleton, which is changing the shape of the membrane. What is membrane potential? How do cells create membrane potential? How is membrane potential used? Membrane potential is an electrical charge across a membrane that creates a voltage difference. How is it created? Through cells expending energy to pump ions across their membranes. A key example, and this is a preview of a topic that we'll get into in Unit 3, is how mitochondria pump protons from the mitochondrial matrix to the intermembrane space. That gives the intermembrane space a positive charge relative to the matrix. It has all these protons inside of it. And that creates an electrochemical gradient, a voltage difference. And that pulls protons through this channel, it's called ATP synthase, back into the matrix, and that powers the creation of ATP. Some other examples, chloroplasts do pretty much exactly the same thing. And there's also a 70 millivolt charge across the membranes of nerve cells. That makes nerve impulses possible, and that's how I am able to think and teach biology, and how you are able to learn it. Is biology mind-blowing or what? I want to acknowledge how difficult and complex some of these concepts can be, and I want to encourage you to go to learn-biology.com, and with a free trial you can do the tutorials and you can use our unit reviews, and it's going to really help you to get on top of this material, setting you up for success on your unit test or the AP Bio exam. Tonicity and osmoregulation. That's the College Board's term. I wish that was just called osmosis and its consequences. So let's start by defining what osmosis is. It's the diffusion of water. Water diffuses like everything else, and that means that water is going to move from higher to lower concentration. Water is the solvent of life, so we have to pay attention to how it moves. Water flows down its diffusion gradient, just like everything else, from hypotonic to hypertonic. Those are essential terms to know. Hypotonic means relatively more water and relatively less solute. And these terms are not absolute. They're always relative to another solution. So in this diagram, this side is hypotonic. It has a higher percentage of water molecules. It has a lower percentage of solute molecules. This side is hypertonic, and that means that it has relatively less water, a lower percentage of water molecules, and a higher percentage of solute molecules. But that's this compared to this. Now, what is that going to do? That's going to have water move down its concentration gradient, which means it'll move from here to here. After osmosis takes place, it'll look like this over here. This level will go down. This level will go up. The water actually is getting pushed up on this side. That's called osmotic pressure. It has enormous consequence. And the thing for you to memorize is that water diffuses from hypotonic to hypertonic. Here's a fabulous and fun example. A student places a gummy bear in a cup of water overnight. The next day, the gummy bear has expanded. Explain. Well, the gummy bear is mostly sugar. The water in the cup is 100% water. Water always flows from hypotonic to hypertonic. So water is going to flow from the hypotonic solution in the cup into the gummy bear. The gummy bear will expand because of osmotic pressure. Osmosis in plants. Use the principles of osmosis to explain each of the images below. On the left side, the cell is hypotonic to the environment. Water leaves the cell, the membrane peels away from the wall. That's also known as plasmolysis. The vacuole shrinks and the plant wilts. How could the environment be hypertonic to the cell? How could the cell be hypotonic to its environment? This environment has to have, for example, a lot of solutes dissolved in it so that it has a lower water concentration than is in the cytoplasm. That'll cause water to go from the hypotonic cell to the hypertonic environment. In the middle, the cell is isotonic to its environment. That's a new term. Isotonic means that the solute concentration and the water concentration are the same on both sides of the membrane. So water enters and leaves and enters at the same rate. And on the right side, the cell is hypertonic to its environment. So water flows into the cell. That osmotic pressure in the case of plants is called turgor pressure. It causes the vacuole to expand. It pushes the membrane against the cell wall. And this is a good situation for plants to be in. It makes them full, firm, happy. They're not wilted. Osmosis in animal cells. Use the principles of osmosis to explain each of the images below. Cell number 1. The cell is hypotonic to its environment. Water leaves the cell. Water always flows from hypotonic to hypertonic, so the cell shrivels up. In situation number 2, the cell is isotonic to its environment. Water enters and leaves the cell at the same rate. This is an important condition for animal cells because they have a membrane but not a wall. Cells that are kept in a tissue culture need to be kept in an isotonic solution. And in situation 3, the cell is hypertonic to its environment, and therefore water is flowing into the cells, and because there's no cell wall, the membrane can't really stop the inward force, and the cell will ultimately burst. Explain the function of the contractile vacuole in freshwater protists such as paramecia. A protist is a eukaryote that's not a plant, an animal, or a fungus. Protists in freshwater are hypertonic to their freshwater environment. That means that the cytoplasm has more solutes dissolved in it than the lake or stream that they're swimming in. And as a result, water moves into these cells by osmosis, flowing from hypotonic to hypertonic. To osmoregulate, that's a new term. Osmoregulate means to regulate your osmotic balance. The contractile vacuole, that's this organelle over here, fills with water and then contracts to expel water from the cell. If the environment becomes more hypertonic, then the cell can adapt by decreasing its rate of contractile vacuole contraction and do the reverse in more hypotonic environments. Describe the structure of leaf stomata. Stomata are pores that are on the underside of a leaf. If you took a leaf and you did a microscopic cross-section, you'd see layers of cells. The top layer and the bottom layer are responsible for waterproofing the leaf, and they're covered with wax. But nevertheless, the leaf needs to be open to its environment in order to exchange gases and other material. Each stoma, which is singular, stomata is plural, is formed by 2 guard cells. So here's one guard cell, here's the other guard cell. And when there's sufficient water, these guard cells buckle outward like that, and that creates a pore that allows carbon dioxide to enter the leaf for photosynthesis, but also allows water vapor to escape. These stomata can be regulated. They can close in response to environmental cues, including water stress, and then open when water is abundant. Explain how guard cells are regulated in order to open and close the stomata. When water is available, cells adjacent to the stomata pump potassium ions into the guard cells. So we're talking about these cells and these cells, the ones that are adjacent to the guard cell, and they're represented like this over here. So the pumping of potassium makes these guard cells hypertonic to the adjacent cells. Water follows by osmosis, causing the guard cells to buckle and open up. When water is scarce, the pumping stops, potassium ions flow out of the guard cells, and water follows by osmosis, causing the stomata to close down, to revert back to this form. What is water potential? Water potential is a concept that's really closely related to osmosis and osmotic pressure, but it's more quantitative. The symbol for water potential is this Greek letter psi. So water potential is a measurement of water's tendency to move from where it is to where it's not. Adding solute decreases a body of water's water potential. Adding pressure increases water potential. And water will flow from areas of higher water potential to areas of lower potential. So if you look at this device over here, it's called a U-tube, and these two parts are separated by a membrane that's permeable to water, but not to the solute. So here we are, we're adding solute to this side of the U-tube. That makes this side hypotonic, but that's another way that you can say it has higher water potential, and water is going to move from higher water potential to lower water potential. That's going to force up the level of water over here. That's going to lower it over here. Could you have explained this entire thing with just hypotonic and hypertonic? Well, I just did, but you'll see that water potential is more quantitative, and it allows for other factors to be included, such as pressure. Explain the formula for water potential. Here it is over here. In plain English, it's water potential equals solute potential plus pressure potential. So psi is water potential, psi sub s is solute potential. And remember that adding solute to water decreases its water potential. Psi p is pressure potential, and adding pressure, like if you pressed on a syringe, that increases water potential. So let's look at these two examples over here. So we've added solute to this side of the U-tube. Pressure potential is 0. There's nothing pressing down on either side, but adding solute lowered the solute potential over here to negative 0.23 megapascals, and that's the unit that's used for pressure. So the total water potential over here is minus 0.23 megapascals. The water potential over here is just 0. There's no pressure, there's no solute, so water moves from higher water potential to lower water potential. And that's why it's moving from this side of the U-tube to this side. In this example, we have a potato cell or potato tissue that's been placed in water. And so again, there's no pressure on either side, it's just an open container. But the cell has a negative 1.0 megapascal solute potential, so the total water potential is negative 1.0. This is just water, so the water potential is 0 over here. Water moves from the beaker into the cell, which has a lower water potential, and that's causing water to flow into the cell. It's causing the cell to expand. All of these things related to the cell wall. In a typical lab in AP biology, you put potato tissue into water or solutions of various concentration, and you can see what happens, and you can actually quantify it based on water potential. That's what water potential is used for. You are a hero for reviewing this much of Unit 2 of AP Biology. That is phenomenal. Now what I want you to do is continue your hero's journey by going up to learn-biology.com and doing the Unit 2 review that I've set up for you. It's got flashcards, quizzes, multiple choice, FRQs, and tap challenges. You will learn so much. You'll feel so confident for your next test or for the AP Bio exam. Unit 2 is a unit for which I have a lot of great music, and I want to encourage you to check out the Cell song, my Membrane song, and my Osmosis song. And now head on over to the next video in this series where we'll review all of Unit 3. See ya.